Hi everyone, this is the pond water organisms lab. So in this lab, I'm just going to talk about how we look at pond water organisms, and we're going to talk about wet mount and different microscopy techniques. Okay, to start off, so just some background, pond water has many free living organisms. It has all types of organisms in it. It has so many different conditions that we see everything growing. So we see prokaryotes, we see lots of bacteria or prokaryotes, we see a lot of bacterial cells there, mainly cyanobacteria. Cyanobacteria are bacteria that do photosynthesis. And there are a lot of eukaryotes. So there's mosquitoes, there's algae, there's arthropods, there's worms, there's rotifers, there's everything. The take-home message I want you to get is that pond water has everything. And here is a collection of pond water. If you look at it with your eye, you can see there's a lot of things. Now, seeing that I can see a lot with my naked eye, most things in pond water are eukaryotes, but there are a lot of bacteria, and obviously you can't see bacteria with your eye. You would need a microscope. So with looking at pond water organisms, you're going to learn a technique called wet mount technique, and I posted a video that shows you how you do wet mount technique. Wet mount technique is a technique that allows us to make slides of living organisms. That is the benefit of wet mount. Basically, you don't do anything to harm the cells. You just take a little bit of your cells, you put them on a slide, you put a cover slip, and that's it. The benefit of these is that you're looking at living organisms. There's no staining, there's no heat, and you can use this method on pond water organisms, meaning no staining, because a lot of pond water organisms are colorful. Like if you look at them, there's a lot of color inherently in there, and motility can be viewed. Motility can be a disadvantage as well, because if you're trying to focus on a structure, like if you're a researcher and you're looking at a specific structure, you may not want it to be motile, but it's an advantage and a disadvantage. But the main advantage is that your cells stay living. And another disadvantage is wet mount technique does not work on transparent organisms. So if you have transparent organisms, you either need to use special microscopy, which I will talk about, or you need to stain your cells, because otherwise they're clear. So just keep in mind that with this, you're gonna see all types of organisms. So if you guys are actually doing this in lab, everyone would have a different slide. You'd see algae, protozoa, insects, a mosquito larva, worms. This here is a good representation of what a pond water slide looks like. You just see everything in it. And contrast with pond water organisms. So most pond water organisms are colorful, and we can see that's why we're doing wet mount with it. But a lot of organisms in general are transparent, meaning they're clear, and they're very hard to see. So the way that we would see transparent organisms is we have to create contrast. You can create contrast either by doing staining techniques in lab, or you can use special microscopy. I'm going to talk about two different contrast microscopy types. One is the fluorescence microscope. If you naturally have fluorescent organisms, so within my pond water here, I have naturally fluorescent organisms, and I use fluorescence microscopy, you'll see fluorescence. So that's one way to see fluorescent organisms. I cannot see fluorescent organisms with my naked eye. I need UV light to shine on them using a fluorescence microscope. Another good microscope technique, which is the way to go with transparent organisms, is called dark field microscopy. Dark field microscopy, what it does is you create a dark background so that you can see clear organisms. Because clear organisms on a clear background, you don't see anything. But if you have clear, transparent organisms and you would do a dark background, you can see them. That's what dark field microscopy is. Here, so if I have my microscope here, I don't want to focus too much on this because I do talk about it in the microscope lecture. So here is my microscope. Let me just make this a little bit bigger. Okay, so here's the microscope. There's a condenser in the microscope. We can put a dark disc in there, and that's how you get dark field microscopy. So it's very easy, and you get these really nice images. And then finally, what I want you to focus on from this lab is to know what pond water organisms look like under the microscope. It's basically a mixture of a lot of things. Know that you're looking at eukaryotes mainly, because in this lab, I don't ask anyone to go to oil immersion. Most of the organisms you can see at low magnifications. know why dark field and fluorescence microscopy are used, and that's mainly because our organisms are transparent. And keep in mind that we only are able to see bacteria or prokaryotes at 1000x total magnification or 100x just regular magnification because they're so small. And then finally, I want everyone to know the advantages and disadvantages of the wet mount technique that we talked about. So just keep all these things in mind.